Look at your hand. It looks clean, right? But at this very second, you are carrying more microscopic organisms on your skin than there are people on the entire planet. Welcome to the Invisible Empire. Microbes, or microorganisms, are the oldest, most successful life forms on Earth. They make up the massive foundation of our biosphere. They live inside boiling volcanic springs. They thrive miles beneath the frozen Antarctic ice. They are even inside your gut right now, helping you digest your last meal. But what exactly is a microbe? How are they built? And how do scientists organize billions of different invisible species into neat, understandable categories? Today, we are stripping away the mystery. We are going to zoom past the limits of human sight to explore the incredible anatomy of microbes and break down exactly how we classify them. By the end of this video, you will see the world in a completely different way. Let's dive in. Microbes are the oldest and most successful life forms on Earth, dominating the biosphere for over 3.5 billion years. They thrive in extreme environments, from boiling volcanic springs to deep Antarctic ice. In the human body, trillions of microbes actively aid in digestion and essential biological processes. These foundational organisms support all life on Earth through several key mechanisms. Biochemical powerhouses. By generating oxygen, microbes transformed Earth's early atmosphere to support more complex life. Extreme survivors, known as extremophiles, they survive in conditions with no sunlight, intense pressure, and extreme temperatures. The human microbiome. The human gut relies on a symbiotic community of bacteria to extract nutrients and synthesize vital vitamins. Before we can look at individual microbes, we must understand the grand division of all cellular life. Every living cell on Earth fits into one of two categories, prokaryotes or eukaryotes. Think of a prokaryote like a simple, open-concept studio apartment. Everything is in one room. There are no walls dividing the kitchen from the bed. In a prokaryotic cell, there is no nucleus. The DNA just floats freely in a concentrated puddle called the nucleid. There are no complex internal compartments. It is basic, efficient, and built for speed. Bacteria and archaea belong here now. Think of a eukaryote like a multi-room luxury mansion. It has specific walls and doors separating the kitchen, the bedroom, and the living room. In a eukaryotic cell, the DNA is safely locked inside a heavily guarded vault called the nucleus. It also contains specialized compartments called organelles, like mitochondria for power, and the endoplasmic reticulum for manufacturing. Fungi, algae, protozoa, and human cells belong here. This fundamental difference in structure dictates how these microbes grow, reproduce, and survive. All living things are made of cells, which are broadly divided into two main types, prokaryotic and eukaryotic. Prokaryotes are simple, ancient, single-celled organisms like bacteria that lack a nucleus. Eukaryotes are larger, more complex cells that house their DNA in a true nucleus and contain specialized, membrane-bound organelles. The fundamental differences between the two cell types come down to size, internal organization, and how they store genetic information. Nucleus. Eukaryotes contain a membrane-bound nucleus that protects their DNA. Prokaryotes do not have a true nucleus. Their DNA floats freely in a concentrated region of the cytoplasm called the nucleid. Organelles. Eukaryotic cells contain membrane-bound organelles, like mitochondria, the endoplasmic reticulum, and chloroplasts, that compartmentalize functions. Prokaryotic cells do not have these membrane-bound structures, though they do contain ribosomes to synthesize proteins. Size. Eukaryotic cells are significantly larger, typically /10 to /100 times the size of prokaryotic cells. Organisms. All prokaryotes are unicellular, belonging to the domains bacteria and archaea. Eukaryotes can be unicellular, like yeast and protists, or multicellular, like plants, animals, and fungi. Despite their differences, both types of cells share essential features that allow them to function as the building blocks of life. All cells, both prokaryotic and eukaryotic, contain DNA, the genetic material that holds the instructions for life. Plasma membrane, an outer barrier that controls what enters and exits the cell. Cytoplasm, a gel-like substance filling the interior of the cell. Ribosomes, tiny structures responsible for building proteins. Prokaryotes were the first kind of organisms to evolve. They are single-celled organisms that don't have a well-defined nucleus and also lack other membrane-bound organelles such as mitochondria. The genetic material in prokaryotic cells exists within an irregularly shaped area of the cell known as the nuclid. All water-soluble components within prokaryotes such as DNA, proteins, and metabolites are found in the cytoplasm, enclosed within the cell membrane and not in separate compartments of the cell. However, bacteria have protein-based microcompartments that behave like basic organelles within protein shells. The two types of prokaryotic cell are archaea and bacteria. Prokaryotes have a cytoplasm, plasma membrane, DNA, and ribosomes, but no nucleus. They have a cell wall that offers an additional layer of protection, prevents dehydration, and maintains the shape of the cell. The cell wall is made of peptidoglycan. Some prokaryotic cells also have a polysaccharide layer, referred to as a capsule, that enables specific attachment of the cell to certain surfaces. Prokaryotes have a plasma membrane, which separates intracellular components from the external environment. Prokaryotic cytoplasm is made up of a gel-like substance called the cytosol, in which cell components are contained. As is the case for all cells, bacteria have a cytoskeleton, a network of filaments and tubules that provides structural support and maintains the shape of the cell. The cytoskeleton also forms a whip-like, external structure called the flagella, which the cell uses to move from one place to another, locomotion. Thin, hair-like structures called pili are also found in some bacteria, which aid in adhesion to other bacteria or host cells. Some prokaryotes have intracellular membranes such as vacuoles or other systems with specific metabolic properties and play a role in processes such as chemolithotrophy or photosynthesis. Some species have carboxysomes or gas vacuoles in the cell, which are carbohydrate-containing microcompartments that play distinct physiological roles. Archaea are single-celled microbes that were initially classified as bacteria and called archaebacteria. Later, it was found that archaea have unique properties that differentiate them from bacteria and eukaryotes. Instead of the fatty acids found in bacteria and eukaryotes, archaea have membranes made of ether-linked lipids, isoprene chains, that provide immense stability. Archaeal walls do not contain peptidoglycan, which is a hallmark of bacterial cells. Instead, they rely on different sugars, proteins, or pseudopeptidoglycan. Although similar in shape and size to bacteria, archaea have genes and metabolic pathways that are more similar to those of eukaryotes. Other biochemical aspects of archaea, such as their dependence on ether lipids in the cell membranes, are unique. Reproduction in archaea is asexual and achieved through fragmentation, binary fission, or budding. Archaea have unique metabolic capabilities, such as methanogenesis, producing methane gas, which is a process no bacteria or eukaryote can perform. Archaea are abundant in oceans. They are thought to play key roles in the carbon cycle and nitrogen cycle. One example of useful archaea is methanogens, which are found in the human gut, where they aid digestion. Another example is extremophile archaea, which have enzymes that are used in biotechnology on account of their ability to withstand extremely high temperatures. So, we have laid out the ultimate biological blueprint. You now know exactly how a bacterium is armed, how its cell wall acts as a shield, and how it is built from the inside out to conquer almost any environment on Earth. But this is just the surface of the battlefield. How does that tiny molecular outboard motor, the flagellum, actually spin at 100,000 revolutions per minute without melting? How do those hidden loops of DNA called plasmids share biological cheat codes to make bacteria completely immune to our strongest antibiotics? And what happens when we zoom even deeper into the internal machinery that keeps these single-celled powerhouses alive? We are stopping right here today because the deep dive into these microscopic machines is so wild, it deserves its own spotlight. In the very next part of this series, we are cracking open these bacterial cells to explore their structures and organelles in extreme, moving detail. You will see exactly how they fight, how they move, and how they operate like fully automated microscopic factories. Trust me, you do not want to miss it. Smash that subscribe button right now, Turn on the notification bell so you get alerted the exact second part 2 drops, and drop a comment below. Which bacterial structure do you want us to dissect first? Until next time, keep exploring the invisible world. See you in part 2.